Hello, everybody. First of all, I'd just like to say thank you to the organizing committee for giving me the opportunity to present some of my research, which really builds upon our previous findings that somatosensory circuit dysfunction is present within SYNGAP1 pathogenesis. Now, part of the lab's research topics have moved a little bit away from the cellular, molecular, and circuit level analyses, and have started to move towards the systems level. So here I'll be presenting some new data on how we think distant brain regions may be modulating sensory perception and detection, leading to altered behavior and cognition. So, as we know, the perception, processing, and integration of sensory stimuli is an integral part of our daily lives. Whether we're conscious of it or not, these types of computations are constantly being performed within our brains and are required to perform pretty much any type of goal-directed behavior. Here, neurons within the periphery act to extract information from the external world. This information gets transmitted to the brain where it is processed, integrated, filtered, all in an attempt to form a coherent representation of the external world, which then we can use to generate some sort of behavioral response. Now, over the last decade or so, it's become well appreciated that sensory processing deficits are fairly common within neurodevelopmental disorders. And this usually manifests itself in a number of different flavors. Rather, individuals are hypersensitive to sensory stimuli and they engage in these aversive types of behaviors. Or some individuals may be hyposensitive and engage in sensory seeking types of behaviors. Additionally, NIMH has recently added the sensory motor system domain to the RDoc matrix, whose main goal is to really provide some useful information about the basic biological and cognitive processes that lead to mental health and illness. And then us as basic researchers and clinicians can use this matrix to form some specific hypotheses about how and where these neurodevelopmental disorders come about. So it's been known for the last century that through works by Yerkes and Dodson that arousal can affect performance. And it follows this inverted U-shaped curve with low and high arousal states resulting in a reduction in performance, and this middle arousal state resulting in high performance levels. Now, the interesting thing is that sensory processing also follows this inverted U-shaped curve when it comes to arousal, with the mid-level arousal state providing the optimal sensory processing kind of state. Now, it can be very difficult to measure arousal in the clinical setting as well as during basic experiments. But one proxy measure that we can use is Pupil Dynamics. Now, Pupil Dynamics offers a quantitative, non-invasive, and potentially translational biomarker for detecting abnormalities in the neural activity involved in adapting the arousal system. And it's been said that the eye is the window into the soul. And if this is true, then I think the pupil is the window into the mind. Now, the pupil can respond to a number of different things, including light, emotion, and cognitive load. But I'm going to be focusing on these two last topics here. I should also mention that pupil diameter, so dilation, is indicative of the arousal state, and constriction is more kind of in the less aroused state. Now, there's been a lot of work to try and disentangle the circuitry that's involved in modulating arousal and pupil dynamics. And what's come about is this area called the locus coeruleus seems to be really important, as it has a dense network of noradrenergic fibers that can release norepinephrine into a number of brain regions, including the cortex, to modulate arousal and therefore pupil diameter. So I know that this background information has probably gone over a number of times, so I'm going to try and keep this short and sweet. Basically, the SYNGAP1 gene is a high-risk locus for a number of neurodevelopmental disorders, a number of different isoforms that can be expressed depending on alternative splicing that is thought to promote different types of biology and pathophysiology. The protein itself is a core postsynaptic density protein involved in the NMDA receptor signaling complex, and its main role is to be a negative regulator of RAS-ERK signaling. But I think it's been recently shown that it's involved in other cellular processes that aren't, strictly limited to dendritic spine biology. So something that's kind of at the forefront of the research. Now, de novo loss of function mutations resulting in SYNGAP1 haploinsufficiency is known to cause a specific form of intellectual disability with epilepsy known as MRD5, and there are a number of cognitive and behavioral deficits associated with these types of mutations. Now, in the lab, We use a construct and face-valid mouse model for SYNGAP1 haploinsufficiency, which is a SYNGAP1 heterozygous knockout mouse. And work from our lab and others have shown that loss of just one copy of this gene results in a whole plethora of defects. And our favorite that we like to study is sensory processing. Now in 2018, we teamed up with Bridge the Gap Foundation, and we were able to mine through the SYNGAP1 patient registry, and we found that there was a high proportion of individuals that suggested that there were sensory abnormalities related to touch, as well as blunted responses to pain. So we spent a couple years working on this and trying to understand some of the deficits and how these sensory processing deficits come about. And what we found was that when we looked within the somatosensory cortex, the brain region that's involved in detecting and perceiving sensory touch, that in response to sensory touch, these neurons produce less activity. These neurons were smaller in size and had fewer dendritic spines. They had a reduction in connectivity, and in an instrumental learning task that required the detection of a sensory stimulus, that the heterozygous animals had a difficult time learning this task. So we gave this kind of real detailed circuit level analyses, but we want to kind of move beyond this specific region and investigate other mechanisms, circuits, and regions at the systems level to gather a better understanding of how altered sensory perception affects cognitive function. So the aim of this set of experiments was to really determine if SYNGAP1 mice have alterations in light-independent pupil dynamics in response to salient sensory stimuli. The rationale is that pupil dynamics offer a quantitative and non-invasive proxy measure of arousal, which is known to promote sensory process and reward learning. So our main hypothesis was that pathogenic SYNGAP1 mutations impair neural activity involved in adapting the arousal system that typically promote cognitive functions. So how do we go about investigating sensory touch in these rodents? Well, we use the Whisker system. Remember that mice are nocturnal animals with poor vision, therefore they used their whiskers or specialized hairs on their face called vibrissa to scan the external world in a similar fashion that we may use our fingers in the dark to determine what an object is and where it may be located. So our go-to task is this whisker-dependent instrumental learning task. It's a go, no-go task. And basically we just train animals to associate a passive whisker deflection with the availability of a water reward, and reporting this passive whisker deflection by licking a lick sensor. So as the name suggests, there are both go trials and no-go trials. On go trials, the whisker is stimulated. If the animal licks, he gets a water reward. It's considered a hit. If he doesn't lick, it's considered a miss. On no-go trials, the whisker is not stimulated. So these are basically catch trials. And if the animal withholds its lick, or if the animal does lick, it's a false alarm. And if he withholds, it's a correct rejection. But he does not get a water reward for this correct rejection. Now, animals do about 300 trials in a given session, 1 session per day, and they do about 20 sessions throughout the training period. And we have started to record pupil dynamics in some of these sessions, namely session 0, so when the animals are completely naive, on session 10, when the animals have learned a little bit, and then on session 21 to see what the pupil dynamics look like after training. So I'm going to go through the behavior data first. What I'm showing here is the fraction of total trials correct as a function of session. We think that when the animals reach over 70% of the total trials correct, that they've reached expert performance levels. They also need a discrimination index of over 1.1. The D prime score is just a measure of how well the animals can discriminate between the go trials and the no-go trials. And so we also train different cohorts of animals on varying stimulus intensities, with the idea that if the head animals have a difficult time learning, and it's strictly due to sensory detection and perception, if we're able to really ramp up the stimulus intensity, we should be able to overcome these deficits and get the animals to learn at a similar level to the wild types. So we started with the low stimulus intensity. Both genotypes, the wild type shown in blue, the het shown in red, both genotypes had a very difficult time learning the task. They didn't really reach their performance levels or on their D primes. So we increased the stimulus intensity and trained another cohort. This time the wild types did fairly well. They have this nice learning curve. The D prime is over 1.1. Now the hets, their learning curve wasn't quite as good as the wild types. And their maximum performance levels were under that of the wild types. And even though they reached their expert performance levels, they still didn't do as well as the wild type animals. Now, remember that we hypothesized that we would see likely a large phenotype here on this mid-stimulus, and then by increasing the stimulus intensity further, we should see the het animals reach the levels of the wild types. And it looks as though that the phenotype got even worse, or even better, however you want to look at it. So again, the het animals still learned the task, but it didn't reach the levels of the wild types. So I think it's quite clear that the SYNGAP1 animals do have problems in this whisker-dependent learning task. However, we don't think that it's strictly due to sensory detection or perception per se. We think that it's likely something else is going on. Hence why we want to look at the arousal system. Now, in order to track pupils throughout these videos, we use this machine learning algorithm software called Deep Lab Cut, in which we have to manually label a number of frames using these 4 labels here over the pupil. We train the neural network, and once we have a good enough network, we can actually run through all of our videos, and the program and algorithm are going to automatically detect the pupil, and we can output traces of the pupil dynamics, shown here. So the data I'm going to show you is for the high stimulus on days 0, 10, and 21, remember. So what I'm showing here is the percent pupil change as a function of the time from the stimulus. And we can see that in all 3 days, that pupil dynamics seem to increase with learning, for both genotypes. However, the maximum pupil change seems to be reduced in the hets compared to the wild types. When we quantify this data, here I'm just showing a correlation of the percent pupil change as a function of D prime. And so it is positively correlated in both genotypes, although the wild type animals have a little bit better correlation than the hets. And again, this is just the maximum amplitude of the pupil change, showing a nice genotype main effect with the hets having kind of blunted pupil responses. So this suggests that the arousal system may be kind of messed up in the het animals. So if you remember back to the SYNGAP1 patient registry, I also mentioned that there were blunted responses to pain. So this is our first attempt to kind of get into the pain field. And of course, we can't strictly ask the mice, how they feel about a certain painful stimulus. So we're kind of stuck at looking at responses to pain. But a number of questions do arise that are very similar to that of the sensory touch phenotypes that we see is, if there are alterations, is it due to perception or changes in internal state or impaired learning, things of this nature. So these are the types of questions that we want to try and answer. And in order to do this, we developed this novel head-fixed shock paradigm. It's all built in-house, custom made. And basically what it is we head fix an animal to this bar here, and they're able to run on the circular wheel. Now, the circular wheel has these copper bars on it, so it's similar to that of a fear conditioning chamber where we can provide foot shocks. We have a rotary encoder on the bottom to record distance traveled as well as velocity of the animals as they run. We have 3 different cameras to look at the face, pupil, and a kind of a profile view. We also have an ultrasonic vocalization microphone to see if these animals vocalize during this task. Now, basically, we habituate animals to head fixation for 4 days, and then on the 5th day, we put them back in and we provide foot shocks. We give 9 shocks per animal that are roughly 1 minute apart. Each shock lasts 1 second at 0.5 milliamps. Again, we have a number of different data streams to look at, and we're using some machine learning algorithms like Deep Lab Cut and VocalMat to probe for differences in the behavioral outputs between genotypes. So this is kind of a nice video that some members of the lab put together, showing you a number of different views of the animal within the shock wheel paradigm. So when the shock comes on, you're going to see a light come on here. I don't know if you can really see it, but there are a number of different labels that are on this mouse, including on the snout, the paws, the body, the ears, and of course the eyes. And we have the output of this kind of behavioral motion shown here as a function of time, and we have a kind of 2D skeletonized version of this. So this is the type of data that we're getting out of this, and we hope to be able to analyze more of this data, not just the pupil, but we're not quite there yet. So maybe next meeting I'll be able to show you some of these cool behavioral responses. All right, so for the pupil, looking at the wild types, these are just kind of representative traces of the pupil dynamics, and you can actually count each of the shocks determined by the changes in pupil dynamics here. So this is a wild type, this is a het, and right away you can see that the pupil dynamics don't seem to be as strong in the het as compared to the wild types. So we quantified this for all the animals, so percent pupil change as a function of shock number. The hets have a kind of blunted pupil response, and when we collapse the data, looking at animal means for all the shocks, we have this nice effect. So this is the second time in a second paradigm that we've seen these blunted pupil dynamic responses. So as a quick summary, I think it's quite clear that salient sensory stimuli can evoke pupil dynamics, and we can track these quite easily in head-fixed mice, which I think could be translated into the clinical population. We show that regardless of the valence of the stimuli, that is a reward or a foot shock, that is pain, the pupil dynamics are blunted in SYNGAP1 mice. So we believe that the circuitry in controlling arousal appears to be dysfunctional, and we think that this may actually be contributing to the deficits that we see in sensory processing, cognition, and other behavioral abnormalities. Of course, at this time, we haven't yet measured any kind of locus coeruleus activity or any of that, but hopefully that'll be on the docket sometime in the near future. And with that, I'd just like to thank everybody within the Rumbaugh lab, specifically those that were directly involved in these experiments, and of course, Dr. Courtney Miller and members of her lab. Lastly, funding agencies. And with that, thanks for your attention, and I'd be happy to take any questions.